A FASTA file is a text-based format used in bioinformatics to represent nucleotide or protein sequences using single-letter codes. These single-letter codes, or nucleotides, are the basic units that make up DNA. Sequences in the FASTA format begin with a single-line description of the sequence, followed by the single-letter codes representing the sequence data. So with our SumGene2 FASTA file, we can use a tool from the National Center for Biotechnology Information, or NCBI, called the BLAST tool, which stands for Basic Local Alignment Search Tool. Using BLAST, we can take sequences we have and search a large database to find sequences that are related to those that we input. BLAST finds regions of similarity between biological sequences and compares nucleotide or protein sequences to sequence databases and calculates statistical significance. There are several BLAST tools to choose from, including those for nucleotides, proteins, and translated nucleotides. For our purposes with our SumGene2 FASTA file, we'll focus on nucleotide BLAST, or BLAST-N. On the BLAST-N page, we can copy and paste one of our gene sequences into the query sequence box and BLAST. You'll get a page that shows descriptions of the sequences producing significant alignments within the search database. There's a graphic summary tab that provides an overview of the database sequences aligned to our query sequence or the sequence we input. Moving into the Alignments tab, we can choose how to view our alignments. The default is a pairwise view, which shows us how each subject sequence aligns individually to our query sequence. Finally, our taxonomy section shows taxonomic reports in three formats: a lineage report, organism report, and taxonomy report. The taxonomy report summarizes the relationships among all of the organisms found in the BLAST results. Using this report, it's easy to see how many records are found within broad taxonomic groups, such as Mammalia. As you can see, the BLAST tool outputs a large amount of useful information that researchers can use to help identify and learn about a gene of interest, from sequence to literature information. Neat.